Hi team, welcome to Adaptive aka Specific Immunity 2. This video will talk about the B cells and humoral immunity. So we'll be down here, humoral immunity. So humor, they call it humoral immunity because humoral just means like liquid, like the blood. And then the B cells make antibodies that live in the blood, so that's why they call it humoral immunity. So here's our hero of this video, the B lymphocyte. Here's its nucleus. Here's its plasma membrane, which is made of a phospholipid bilayer. And then here's the B cell receptor that will recognize specific shapes as shown in this virus. So this is a virus. These would be the spikes on the virus. And this specific B cell receptor, because remember when we're in this specific immune system, this specific B cell receptor would only recognize this red ball. It wouldn't, this specific B cell receptor would not recognize a triangle. It would not recognize a square, etc. So in a single B lymphocyte, the surface is covered with identical BCRs. Those are B cell receptors. The major function of the B lymphocyte, the cell itself, is to make antibodies. And B lymphocytes are responsible and associated with the humoral immune response. You find these B lymphocytes primarily in the lymph nodes, but you can also find them in the spleen and mucosa-associated lymphoid tissue. You can find them in gut-associated lymphoid tissue, and RALT, respiratory-associated lymphoid tissue. Only a small percentage of these B cells are circulating in the blood, but the antibodies circulate in the blood. That's why it's the humoral immunity. A surface of a B cell could be covered with up to 500,000 identical copies of a B cell receptor. And B cell receptors are made out of an antibody. So if you know anything about antibodies, they have this Y shape, right? So the B cell receptors and then the antibody that those B cells secrete would be almost identical. So here's our cartoon. This is a B cell, so here's the B cells, phospholipid bilayer, and then here's the B cell receptor shaped like a Y, and then this is the transmembrane, so it's going across the cytoplasmic membrane. This would be and then it's hooked onto the external part here. And then this could be just like the tip of a rod bacterium with its epitope. So it's antigen epitope. Remember, it's kind of tricky. This is how kind of sloppy it is, I think, in the nomenclature. So here's the epitope that the B cell receptor will bind, but they call the part of the B cell receptor that the epitope will bind, they call it the antigen binding site. So it could also be called the epitope binding site. I usually just try to say antigen epitope, like it's one word, even though we've pointed out that they're distinct shapes. The inside of the B cell receptor is made of a heavy chain, so this guy, and then the outside of the B cell receptor is a light chain. And then you have these tips here, the variable regions. That means they can recognize a variable amount of epitopes. But I have to point out that if you are a single B cell, you have identical B cell receptors in your plasma membrane, or your cytoplasmic membrane, and those B cell receptors can only recognize this one epitope. So each B cell that you have in your body randomly generates a single BCR, or B cell receptor gene. So that means, without getting into the genetics of it, That means that you have in your body 10 to the 9th or 1 billion different possible B cell receptors in your body. That means that your B cells with their B cell receptors can recognize up to 1 billion different epitope/antigens out there in the environment. So that's kind of cool. And you and your brothers and sisters probably would, and your parents and relatives, would share more of the same B cells and B cell receptors than someone that's not in your family. This is important to note. The B cell receptors are not formed in response to an antigen encounter. So when this B cell encounters an antigen or epitope, say it's part of a bacteria, the B cell receptor is not generated at that point. It's already made. It's already there. So you're already kind of tied down to your own immune system. If your B cells don't have a B cell receptor that can recognize a specific antigen epitope, then that bacteria won't be destroyed. So all of your B cells with all their B cell receptors are already present at birth. Okay, then we talked about this. These variable regions are called antigen binding sites, but they could easily be called epitope antigen binding sites. And these variable regions are complementary to the shape, to the epitope/antigens. So see how this is slanted in here, slanted in here, and then you have this cone, so it would fit in. So for example, if this were the antigen epitope, then that variable region would have to be shaped something like this so that it could accompany that circle. Every B cell has unique B cell receptors. So you have a whole bunch of different B cells in your body, and every B cell has unique B cell receptors, kind of like snowflakes are unique. And so when your mom or your teacher says, hey, you're really special and there's no one else like you, they're right in terms of your B cells and your B cell receptors. But then I pointed out again and again, all the B cell receptors on an individual B cell are identical. Okay, so now in this cartoon, this kind of piece around the epitope would be the antigen, and then this whole big purple thing could, for example, be a bacteria. Or this whole purple thing could be the antigen part, and then this little cone shape is the epitope part. But the epitope is that molecular fragment on an antigen that antibodies recognize. So here's the recognition process. And then you get an immunological response. So we've looked at this cartoon before, the B lymphocyte and its nucleus. This is when the B cell gets activated, it turns into this thing called a plasma cell. So this could be a B cell here. When it's activated, it turns into a plasma cell, gets a buttload of rough endoplasmic reticulum in here, and starts cranking out antibodies, which I usually call ABs, antibodies. So when an epitope, so this red thing here on this virus, that's the epitope, stimulates a B cell via its B cell receptor, which is happening right here, the B cell will respond to make a clone of itself. The clones are then, they start secreting antibodies, and they're called plasma cells. The plasma B cells, so they were B cells, but they differentiated into plasma cells when they started making antibodies. But you can call them plasma B cells too, just as long as you know the difference between a B cell and a plasma cell. Plasma cells make antibodies, and these antibodies are epitope specific. So this B cell, this plasma cell, will start making antibodies that are specific to the original, to a piece of the original bacteria that the B cell receptor, which was stuck here in the previous B cell, oops, I should put it there, responded to. So plasma B cells are descended from activated B cells. So B cell has to get activated, and that's very complicated. We'll talk about that. Plasma cells can make huge quantities of specific antibodies. What I'm saying, they're specific to one epitope / antigen. And that's why I say here, the plasma cells secrete a specific antibody specific for a single epitope. That's why it's the specific immune system. Antibodies are also called immunoglobulins. And the antigen binding sites on the antibody here from a given, the antigen binding sites on antibody arms. So these are the arms here, or the Fab regions. And then there's this hinge region in here, and then there's a stem region. So the antigen binding sites on antibody arms, which would be here, from any given plasma cell are identical to that cell's B cell receptors. So this is the B cell receptor. When it got activated, it turned into a plasma cell. That plasma cell started making antibodies that were identical to that B cell receptor. And then there's other parts of these antibodies, like the variable region of the light chain, the variable region of the heavy chain, the constant region of the light chain, the constant region of the heavy chain here. And then these S's are disulfide bonds that hold the antibody together. So the epitope antigen binding portion here again of an antibody is in this Fab variable region. And then, so that's where you, so in this region, this is where you bind the epitope or antigen of the invader. This part down here, the FC region, so from here to here, this is FC region. That's where you can bind to white blood cells. So this is a free antibody, this is not a cell. If you are a white blood cell, let's say down here, white blood cell, you could have a FC binding region in your plasma membrane. That means that the FC region of an antibody could bind here. B cells, classes of antibodies. So the classes of antibodies, just you can remember there's like an old-timey woman's name, Madge, IgM, IgA, Madge. So D, G, and E spell out a woman's name, Madge. That's how you can remember the different classes of antibodies, IgM, IgG, IgA, IgE, and IgD. So in their DNA, so that's what this cartoon is right here, in their DNA, the plasma cells will attach the gene for the variable region. So, and here's the different variable regions. Oh, the gene for the variable region, sorry, right here. So this is a variable region. So this would be unique to a single B cell, that variable region. Can attach that gene, the variable region of interest, to one of these 5 basic FC or stem region genes to make a complete antibody. And so then these would be all the, this would be the gene for IgM, the gene for IgD, etc. So that's how you get your 5 classes of antibodies. Initially, so when you first start this process of making antibodies, the plasma cells will attach the gene for the variable region of that plasma cell to this region, the mu region or IgM region. So initially, when you're making antibodies in response to a foreign invader, you're making IgM. And then all this complicated genetics, but we're not going to go into the detail of that. So yeah, like I just said, IgM is the immunoglobulin or antibody produced during the very first or initial stages of your immune response to a foreign invader. And then there's this cool stuff. Like if you've taken a genetics class, you can read about this class switching. It's pretty cool, but that's genetics class. So if you are a plasma cell and you've been activated because you know there's a foreign invader there, because you know its antigen epitope is there, the first antibody that you make is IgM. This is a cartoon of IgM. It's basically 5 of these units linked together in a circle. So IgM, it's kind of big. During the initial stages of an immune response, so the foreign invader's gotten in, you've mounted your immune response, they're the most common antibody in the blood. The class of antibody made also depends on the type of invading foreign antigens, the portal of entry, which way did the invading foreign pathogen with its antigens come in. Your body can know which immunoglobulin to make. IgG, so this is a pretty simple one, is next made by the plasma cell after IgM. This is important to note, IgG is the only antibody to cross the placenta. So the fetus is getting some antibodies, but they're only IgG. But IgG is the most common antibody, and it's the longest lasting one. So go IgG. As you can see, it's a single unit, plays a whole bunch of roles in the body. It can easily squeeze out of blood vessels to go into the extracellular space where the antibodies are often hiding outside the blood vessels. So that's cool. IgA is associated with your secretions. So secretions by your mucous membranes, saliva, tears, breast milk, all have IgA. And this is a cartoon of it. Some IgAs are single units, but some are dimers, like this one joined in a J chain. So here would be a single IgA, here's another single IgA, and then it's joined here in the middle by this J chain. And because it comes in breast milk, nursing newborns get IgA from mother's milk. Okay, IgE is the antibody of allergies, and we'll talk a whole bunch about IgE when we do allergies. But basically, here's a cartoon of IgE. Kind of looks like IgG. But the IgE will attach to receptors on eosinophils to trigger release of cell-damaging molecules onto the surface of parasites, particularly worms. So if you are infected with worms and they do a blood cell antibody titer and your then your IgE would be up because it's helping eosinophils kill those worms. IgE can also trigger mast cells and basophils to release histamine, ergo your allergies. Got to take an antihistamine for that. And then there's also IgB, but the exact function is unknown. All right, this cartoon talks about primary versus secondary immune responses. So on the y-axis, here's the antibody concentration in the serum. The days are on the x-axis. There's a primary response cartoon and a secondary response cartoon. So you're first infected, there's this lag period. It takes time to make all your antibodies, but the first antibody you can see is made as IgM, and then IgG is made secondly, and then the total antibody titer is shown here. So the lag period, and then following the second, so here's the lag period in the primary exposure, here's the lag period in the secondary exposure to that same antigen that made you sick the first time. So the lag period following secondary exposure, which is here, it's shorter than the initial lag period. There's an enhanced immune response to subsequent exposures. So that's the secondary response to an antigen. if your body has already been exposed, your body knows that antigen and can mount a secondary response, aka a memory response. And then here's this table kind of shows primary immune response and then the secondary immune response. Okay, so this cartoon shows time is on the x-axis, and then on the y-axis is the presence of SARS-CoV-2 viral RNA in swab samples. So initially here you are exposed to SARS-CoV-2. There it is. You're getting exposed to it through your nose. And then over time, it kind of shows the different antibodies that are made in response to that. So let's look at the scenario. A sick child may have influenza or they might have COVID-19. So they're two different viruses, they're two different infections. They have different treatment options. So the physician asks for an antibody titer test. Then here's the results. So anti-influenza antibodies are primarily IgM, and anti-SARS-CoV-2 antibodies are all IgA and IgG. What's the most appropriate interpretation? So you look here at the graph, it looks at like there's all of them. There's the anti-influenza antibodies, IgG, but then there's also the anti-SARS-CoV-2 antibodies, IgA and IgM. So what does the physician think? The child currently has influenza and, in addition, has been previously exposed to SARS-CoV-2. So at day 14, there's almost no detectable amount of viral SARS-CoV-2 viral RNA by this technique called RT-PCR, real-time PCR. So that's a fun little scenario of a sick child having both infections. So what do antibodies do for us? So we say that B cells get activated and they make antibodies, but then what do the antibodies do for us? So there's 6 ways that antibodies help us. So the first cartoon, antibodies can activate complement, and then complement can do this membrane attack complex formation, and then microbes can be destroyed. So here's a complement pathway. So let's look at this way. We're in the classical pathway with antibodies. It shows here the antibodies are, the specific antibodies are binding to that specific epitope on the bacteria. And then when that happens, you can activate the complement cascade. So these complement cascade proteins, C3b, C3a. But for this cartoon, activation of complement and the MAC formation, those you get a whole bunch of complement proteins, and they can form, the complement proteins form the slats in this portal here. And then you can, when that happens, when you put a hole in this bacteria, it lyses. So it bursts, and then, thanks antibody, you just destroyed the bacteria for us with the help of complement. So this just gives you some details. The Fc variable region binds to the first complement protein and then activates the complement cascade. This is a cartoon of activation of complement. This is structure of IgG. So here's the antibody. You can see, just, I know there's a lot of stuff on here, but you can see the Y, right? And what this shows is how detailed the complement binding to an antibody is. So, for example, C3b and C4b binding region is in the constant light chain. C1q binding region is the constant heavy chain too. But anyway, you don't have to know all these details. It's just that complement proteins can bind to the antibodies in specific places. Okay, what else do antibodies do for us? Number 2, they neutralize invaders. So this cartoon is for neutralization. Here's a bacterium. Here's the antibody that's binding to this specific green triangle, which is the epitope antigen. And in this and then also, so this would be this top guy would be a bacteria. It says bacterium, and then this guy would be a virus with these spike proteins, and the virus and antibodies would bind specifically. So these antibodies would not recognize a square spike on this virus. The antibodies can also recognize just a toxin, so this green triangle toxin, and recognized by antibodies, and then these toxins and proteins become neutralized. When they're neutralized, they can't get into the host cell. The third thing the antibodies do for us is opsonization. So antibodies act as opsonins to coat the surface of the microbe. So these antibodies, this is a single antibody, but you could have more antibodies coating, so making a nice coat on the surface of the bacteria or microbe, and that stimulates phagocytosis. So when this bacteria is covered with antibodies and this phagocyte has an Fc receptor here, then it can bind to the Fc portion of the antibody, and that antibody is bound to a bacteria, and now the phagocyte can engulf the bacteria and destroy it. Antibodies can also allow for this process of killing the microbe by oxidation. So some antibodies, when they bind to the bacteria and the phagocyte, will tell the phagocyte to make a whole bunch of hydrogen peroxide and other oxidants and dump that hydrogen peroxide on the bacteria, and it's gone. The fifth thing that antibodies do for us is agglutination. So here's a bunch of antibodies agglutinating all these bacterial cells. And then if you do this agglutination or clumping, then there's a better chance that this clump of bacteria and antibodies will be phagocytized, or they can get filtered out by the spleen. But either way, bye-bye antibody. Then number 6 is antibody-dependent cellular toxicity. So here's a natural killer cell, natural killer lymphocyte, and it's got this Fc region receptor protein, so it's binding the Fc region of the antibody, and the antibody happens to be attached to an epitope on a bacterial cell. So when you get this link up, then this NK cell gets activated, and it can cause these perforin molecules to form a hole in the bacteria, and it lyses the bacteria. And once these perforins are there to poke a hole, then the NK cell can also pour in this molecule, it's called granzyme, and then granzyme triggers the bacterial cell to do apoptosis or cell suicide. Okay, let's talk a little bit about these things called major histocompatibility complex proteins, or MHC. The function of these MHCs is to present epitopes to T cells. And then we'll talk more about them when we talk about T cells. But there's class 1 MHC proteins. You have class 1 MHC proteins, your own special, like dog tags. Let people know that this class 1 MHC protein is only found on your cells. So class 1 MHC proteins are found on any nucleated cell in your body. So that means they're not found on red blood cells, because red blood cells don't have a nucleus. Class 2 MHC proteins, they are only found in B cells or other antigen presenting cells like a dendritic cell. There's two classes, like I just talked about, class 1 and class 2, and where they're found. B cells, the reason I throw in these MHCs is because B cells are antigen presenting cells and they also have a nucleus. So B cells have class 1 MHC proteins in their phospholipid bilayer or their cytoplasmic membrane, and B cells are antigen presenting cells, so they also have class 2 MHCs. So antigen presenting phagocytes, or the antigen presenting cells, will phagocytose a bacterial invader, chop up the invader, and present these special epitopes of the now deceased bacterial invader in these MHC2 proteins, and then helper T cells will be able to come along and see that epitope of the pathogen and here, and the helper T cells will recognize that with their helper T cell receptors, and a whole bunch of cool immunology and biochemistry and cell biology ensues that we'll talk about later. Well, all right, so let's talk about this process of clonal deletion of lymphocytes. So when you make a B cell, your body makes a B cell or a T cell, and their B cell receptors and their T cell receptors. Your body wants to make sure that those B cell and T cell receptors don't recognize any antigens in your body, because if they do and they mount a response, then that's an autoimmune disease. So B cells and T cells, again, randomly generate through DNA magic the shapes of their receptors on their surfaces. So every population of maturing lymphocytes, so B cells, remember, mature in the bone marrow, T cells mature in the thymus. When these young lymphocytes are maturing, some of their receptors that they generate, so some B cell receptors or T cell receptors, they wrongly match with normal body cells or self cells. Those lymphocytes must die or you'll get an autoimmune disease. So that process is called clonal deletion. So here's a pre-B cell. So it's maturing in the red bone marrow. It doesn't have any B cell receptors. They're calling them membrane IGs, but there's no B cells receptors. Sorry. Now this B cell randomly generates this B cell receptor here sticking out of the cell membrane. If that B cell receptor recognizes self antigens, then that B cell dies. If these B cell receptors do not recognize self cells, then that B cell will survive and become a mature B cell. It will go from becoming an immature to a mature B cell. The body will eliminate self-reactive B and T lymphocytes via this process of clonal deletion. And the way that happens is these autoantigenic cells, these are the ones that, these are B cells and T cells that recognize autoantigens, which they shouldn't. They are clonally deleted using this process of apoptosis, which is just cell suicide, basically. So this is a cartoon. Here's a stem cell in the red bone marrow. So they make these B cells, and they want to become mature B cells. So it shows here if this B cell generated a B cell receptor and it recognized a self or autoantigen on a cell, it dies via apoptosis. If and that's, yeah. Then so I don't know if you can see, I kind of marked it out, but this B cell receptor is doing the recognition of the autoantigens. In this cartoon, check it out. The tip of that B cell receptor is not recognizing these green autoantigens. So it gets to go, be a B cell, go to the spleen. And I think that's it.